Hello, I am Professor Sims, and in this video I will discuss muscle tissue. This is the 8th in a series of 10 lessons held as part of my Anatomy and Physiology 1 course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. In this unit, we will learn about the organization of muscle tissue, the function and structure of skeletal, cardiac, and smooth muscle, how muscles and tendons move the body, how muscles contract and relax, the process of muscle metabolism, how the nervous system controls muscle tension, the connections between exercise and muscle performance, and the development and regeneration of muscle tissue. There are 3 main types of muscle tissue in the body: skeletal, cardiac, and smooth muscle. All muscle tissue is derived from the mesoderm and accounts for more body weight than any other tissue type. Muscles share several key characteristics. These are excitability, contractility, and extensibility. So what that means is they respond to stimuli and their electrical state. They can shorten or pull, they can stretch, and then elasticity means that they can return to their original length after stretching or contracting. There are 3 general types of muscle tissue, like we've said previously. So these are going to be your skeletal, smooth, and cardiac. Skeletal muscle is voluntary and it has long cylindrical cells with multiple peripheral nuclei. Skeletal muscle shows a striped pattern called striations, and it accounts for about 40% of the body mass, and these are the muscles that are attached to bones. Skeletal muscles contract quickly and with short twitches. Smooth muscle is involuntary, consists of short spindle-shaped cells with a single nucleus. Smooth muscle lacks striations and intercalated discs, and it's found in the walls of hollow organs like the digestive, respiratory, urinary, and reproductive tracts, as well as in the blood vessels and the erector pili muscles of the skin. Smooth muscle contracts slowly and maintains long-lasting contractions, playing critical roles in moving materials through the body and regulating blood vessel diameter and blood pressure. Cardiac muscle is found only in the heart. It's involuntary. It has short branched cells with 1 or 2 nuclei. It has striations and special junctions called intercalated discs. It has intermediate speed twitches and is responsible for pumping blood and maintaining blood pressure. So we'll look at each type of muscle in more detail now. First, we'll look at skeletal muscles. These are essential for movement and maintaining posture by continually adjusting to resist gravitational forces. They also stabilize bones and joints, prevent misalignment or dislocation, protect internal organs, especially those in the abdominal and pelvic regions, and they generate heat during contraction, which helps to regulate body temperature. Each skeletal muscle is composed of muscle fibers, blood vessels, nerve fibers, and connective tissue. Layers of connective tissue called mysia surround and organize the muscle, providing structural support. The connective tissue of muscle, collectively known again as mysia, play essential roles in supporting and organizing muscle structure. The epimysium surrounds the entire muscle, providing an outer layer of protection and support. Within the muscle, the perimysium encloses each muscle fascicle, which is a bundle of muscle fibers. Surrounding each individual muscle fiber or muscle cell is the endomysium, a delicate connective tissue layer that helps maintain the fiber's environment. Additionally, an aponeurosis is a broad, tendon-like sheet that serves to attach muscles to bones or other tissues, allowing for the transmission of muscular force. Muscular fibers are long cylindrical cells formed by the fusion of embryonic myoblasts. They contain specialized structures including sarcolemma, sarcoplasm, and sarcoplasmic reticulum, which you can see here in figure 10.4. I'd like to show you a short clip now about skeletal muscle structure that will help to better illustrate these structures and substructures in the skeletal muscle.
Skeletal muscles come in different shapes and sizes, but the main structure of a skeletal muscle cell is basically the same. If we were to take a cross-section of a single muscle, we would find it is covered in a protective sheath or layer called the epimysium. The epimysium protects the muscle from friction against other muscles and bones. It also continues at the end of the muscle to form, along with other connective tissues, the tendon which attaches the muscle to the bone. Each muscle is made up of a number of bundles of muscle fibers called fasciculi. The fasciculi are contained within another connective tissue called the perimysium. Each bundle of muscle fibers contains anywhere from 10 and 100 individual fibers. A large, strong muscle, such as those in the quadriceps group, would have a large number of fibers within each bundle. A smaller muscle used for precision movement, such as those in the hand, would contain far fewer muscle fibers in each bundle. Each muscle fiber is also covered in a fibrous connective layer of tissues called the endomysium, which insulates each fiber. Beneath the endomysium is the sarcolemma, which is the muscle fiber's cell membrane, and beneath this is the sarcoplasm. This is a gelatinous fluid which contains glycogen and fats for energy, as well as mitochondria, inside which the cell's energy is produced. Each muscle fiber itself contains cylindrical organelles known as myofibrils. Each muscle fiber contains hundreds or thousands of myofibrils, which themselves are bundles of proteins called actin and myosin. These run the length of the muscle fiber and are important in muscle contraction. Surrounding the myofibril, there is a network of tubules and channels called the sarcoplasmic reticulum, where calcium is stored, which is also important for muscle contraction. Each myofibril can be broken down into functional repeating segments called sarcomeres, which are the basis for muscle contraction theory known as sliding filament theory.
Okay, back to the textbook. This is figure 10.5 showing the sarcomere, and this is the fundamental contractile unit of a muscle fiber. The sarcomere is responsible for the striated appearance of skeletal muscle. It's defined by Z lines and contains distinct regions. So these are the Z lines. Z line here. The A band is where actin and myosin overlap. The I bands contain only actin, and the H band, the H zone here, contains only myosin. The M line anchors the thick myosin filaments at the center of the sarcomere. Thin filaments are primarily made of actin, along with the regulatory proteins tropomyosin and troponin, while thick filaments consist mainly of myosin. During contraction, myosin heads bind to actin and pull the thin filaments inward, driving the sliding filament mechanism that generates muscle force. Please watch this short clip that shows the sliding filament mechanism and how it works.
If we look at a two-dimensional model of a sarcomere, it consists of actin and myosin. When a nerve impulse arrives at the muscle, it causes a release of a chemical called acetylcholine. The presence of acetylcholine causes depolarization, enabling calcium to be released from the sarcoplasmic reticulum. The calcium binds to troponin, changing its shape, and so moving tropomyosin from the active site of the actin. The myosin filaments can now attach to the actin, forming a cross bridge. The breakdown of ATP releases energy, which enables the myosin to pull the actin filaments inwards, contracting the muscle. This occurs along the entire length of every myofibril in the muscle cell. When an ATP molecule binds to the myosin head, the myosin detaches from the actin and the cross bridge is broken. When the ATP is then broken down, the myosin head can again attach to an actin binding site further along the actin filament and repeat the process. This repeated pulling of the actin over the myosin is often known as the ratchet mechanism. This process of muscular contraction can last for as long as there are adequate ATP and Ca+ stores. Once the nerve impulse stops, the Ca+ is pumped back to the sarcoplasmic reticulum and the actin returns to its resting position, causing the muscle to lengthen and relax.
The site where the motor neuron and muscle fiber meet is called the neuromuscular junction. This is a specialized synapse where the nerve ending comes into close contact with the sarcolemma, allowing the neuron to communicate with the muscle. At the neuromuscular junction, the motor neuron releases the chemical messenger acetylcholine, which binds to receptors on the sarcolemma and initiates an electrical signal in the muscle fiber. When an action potential reaches the end of a neuron, it opens voltage-gated calcium channels, causing acetylcholine to be released into the synaptic cleft. Acetylcholine binds to the receptors on the motor end plate of the muscle cell membrane, opens the sodium channels, and then this triggers depolarization. The This electrical signal spreads through the sarcolemma and travels into the muscle fiber via T-tubules. At the triads, where T-tubules meet the sarcoplasmic reticulum, the signal prompts the SR to release calcium into the sarcoplasm. Calcium binds to troponin, which shifts tropomyosin and exposes binding sites on actin. Energized by ATP, myosin heads attach to actin and pull the thin filament inward, creating contraction through the sliding filament mechanism. This cycle continues as long as calcium and ATP are present. To stop the signal, acetylcholinesterase breaks down the acetylcholine in the cleft. As the action potential ends, the membrane repolarizes, sodium channels close, potassium exits, and sodium-potassium pump restores balance. Calcium is pumped back into the SR, binding sites are covered again, and the muscle relaxes. Muscle fibers rely on several energy sources to keep contractions going. Initially, they use stored ATP, which provides energy for just a few seconds, roughly 10 muscle twitches. Once that runs out, creatine phosphate kicks in, quickly regenerating ATP and extending contraction for about 15 more seconds, or about 70 twitches. As activity continues, muscles shift to glycogen stored in the muscle, using anaerobic glycolysis to produce 2 ATP per glucose molecule, supporting contraction for about 130 seconds or 670 twitches. Though this also produces lactic acid. And for longer lasting energy, muscles turn to aerobic respiration, also using glycogen. But this process involves the Krebs cycle and the electron transport chain, which generate about 36 ATP per glucose molecule and fuel muscle activity for up to 2,400 seconds, or about 12,000 twitches, as long as oxygen is still available. Figure 1012 shows us what this looks like in the muscle fibers. So here you have the resting muscle where ATP is stored, and then as the contraction starts, it is used up in seconds. But if you have creatine phosphate, this is going to produce more ATP, and then this energy will last for up to about 15 seconds. Now after that, you will have to go into using glucose via either aerobic or anaerobic respiration. However, in anaerobic respiration, that's where you get lactic acid in the blood. And this is actually what causes cramping after you've done a lot of exercises and your muscles start to cramp up. That's what's happening, is that your muscles had to use anaerobic respiration in order to keep contracting. Figure 10.12c is showing how anaerobic respiration is happening in the mitochondria of the muscle cells. And this is going to produce by far the largest amount of ATP. About 95% of the energy required for resting or active muscles is provided by aerobic respiration. Muscle fatigue occurs when a muscle can no longer perform at the required level of activity. It can result from ATP depletion, lactic acid buildup, or structural damage to the sarcolemma or sarcoplasmic reticulum. After peak activity, contributing factors include a drop in pH from metabolic acids like lactic acid and also carbonic acid, reduced calcium ion binding to troponin, and altered enzyme activity. Recovery involves restoring the muscle fibrous conditions to pre-exertion levels. While moderate activity may require only a few hours for recovery, intense or sustained exertion can take up to a week for full recovery. The strength of muscle contraction depends on the number of myofibrils and sarcomeres in each fiber. Muscle size increases through hypertrophy and decreases with atrophy. In disorders like Duchenne muscular dystrophy, a lack of dystrophin, which is an essential structural protein, weakens muscles, especially in males, and often leads to mobility loss, respiratory failure, and even early death. Treatments for DMD focus on increasing eutrophin in order to compensate for dystrophin deficiency. After exercise, the body experiences an oxygen debt. The extra oxygen required to restore ATP and creatine phosphate levels, convert lactic acid to pyruvic acid, and then to help the liver convert lactic acid into glucose or glycogen. So this is why breathing remains elevated even after activity has stopped, because your body is deprived of oxygen. Isotonic contractions occur when muscle tension increases in enough to move a load, and once movement begins, the tension remains relatively constant. There are two types of isotonic contractions: concentric contractions, in which the muscle shortens during contraction, and eccentric contractions, in which the muscle lengthens during contraction while still generating force. Isometric contractions occur when a muscle generates tension without changing its length. In this type of contraction, the muscle does not shorten or lengthen, and no movement occurs at the joint. Instead, the muscle fibers produce force to resist an opposing force or maintain a fixed position, such as holding a heavy object still or maintaining your posture. A muscle produces the most tension when stimulated at its optimal length. Once calcium is released from the triads, the entire fiber contracts fully. There's no way to partially activate a sarcomere. It's very all or nothing. A motor unit includes all muscle fibers controlled by a single motor neuron. Smaller motor units provide more precise movements. And since fibers from different motor units are interwoven, increasing the number of active motor units, which is called recruitment, allows for smooth increases in force. During long contractions, motor units take turns working through asynchronous summation, so they get brief rest periods and can recover faster. Before we go into more detail about how muscle fibers are stimulated, please watch this clip about how motor units work.
The central nervous system controls every skeletal muscle through an alpha motor neuron. The alpha motor neuron cell body is located in the spinal cord at the ventral horn. The axon exits through the ventral roots and inserts into muscle fiber. A motor neuron axon branches many times after entering the target muscle, and each branch makes its way to different muscle fibers and forms a small cluster of terminal branches. When a motor neuron fires, all the muscle fibers in the motor unit contract at the same time. The size of a motor unit varies from just a few fibers in the eye muscles, precise movements, to over 1,000 threads in the large leg muscles, powerful movements. The all or none law. This principle states that if a single motor unit receives a stimulus of sufficient intensity to bring forth a response, all the muscle fibers within that unit will contract at the same time and to the maximum extent. If the intensity or duration of the stimulus is increased, the height of the impulse will remain the same. The nerve fiber either gives a maximal response or none at all. However, the muscle can regulate its strength by activating only the required number of motor units needed for a specific action. For example, if a trainee performs squats without additional weight, a small number of motor units will be recruited together. But when a trainee does the squat with heavy weights, more motor units will be required to complete the exercise.
A twitch is a single stimulus contraction relaxation sequence in a muscle fiber and can vary in duration depending on muscle type, location, internal or external environmental conditions. The tension generated by a single muscle twitch can be recorded using a myogram, which tracks tension over time. A twitch consists of 3 phases: the latent period, when calcium is released but no tension is produced yet; the contraction phase, when cross bridges form and muscle tension increases; and the relaxation phase, during which calcium is reabsorbed, actin binding sites are covered, and the muscle fiber returns to its resting state. If fibers are stimulated while a previous twitch is still occurring, the second twitch will be stronger. This response is called a wave summation because the excitation-contraction coupling effects of successive motor neuron signaling is summed or added together. If the stimulus frequency is so high that the relaxation phase disappears completely, contractions become continuous in a process called tetanus. There's different types of tetanus. When a muscle is stimulated repeatedly with brief periods of relaxation in between, it enters incomplete tetanus, where tension increases with each stimulus but does not reach its maximum. If the stimuli occur rapidly enough that there's no time for the muscle to relax at all, then that is complete tetanus, and it results in a sustained maximal level of tension. And then finally we have trepe, or the staircase effect, where muscle contractions gradually become stronger with repeated stimulation, provided that each stimulus follows a complete relaxation period. Even when a muscle isn't actively contracting, some motor units remain slightly active, creating a low level of tension known as muscle tone. This resting tone helps maintain posture and readiness for action. When muscle tone is abnormally low, this is called hypotonia, and in this state, muscles become limp and weak, often due to nerve damage or central nervous system disorders such as polio. In contrast, excessively high muscle tone is called hypertonia, and it leads to stiffness or spasticity, which is commonly seen in conditions like Parkinson's disease or after certain strokes. Muscle fibers are classified into 3 types based on contraction speed and ATP production. Contraction speed is influenced by myosin ATPase activity, which regulates cross-bridge cycling. Slow oxidative fibers with their high mitochondrial content produce more ATP and sustain actively longer, though they generate less force, contract more slowly, and rely on aerobic respiration. They still resist fatigue fairly well. Fast oxidative fibers balance speed, force, and endurance. They contract more quickly, primarily use aerobic respiration, but can shift to anaerobic metabolism when needed, and they offer moderate fatigue resistance. Fast glycolytic fibers contract rapidly, depend on anaerobic glycolysis, and fatigue quickly due to the limited ATP yield from anaerobic pathways. Most human skeletal muscles contain a mix of all 3 of these fiber types, with proportions varying depending on the muscle's primary function. This table here helps to summarize the properties of the different types of muscle fibers, including cross-sectional diameter, time to peak tension, contraction speed, fatigue resistance, glycolytic enzymes, and also ATP substrates. Physical training can alter both the appearance and performance of skeletal muscle. Muscle growth occurs through hypertrophy, where structural proteins are added to increase cell diameter, not by generating new muscle cells. In contrast, atrophy happens when these proteins are lost and reduced muscle mass results. Endurance exercise primarily engages slow oxidative fibers, which are highly resistant to fatigue, and it enhances aerobic capacity by increasing mitochondria, myoglobin, and promoting angiogenesis to improve oxygen delivery to muscle tissue. These adaptations improve the ability of muscles to sustain prolonged activity. In contrast, resistance exercise targets fast glycolytic fibers, stimulating the production of myofibrils, which increases muscle fiber thickness and leads to hypertrophy. And this improves muscle strength and power for short, intense bursts of activity and strengthens connective tissues. While exercise benefits muscle health, performance-enhancing substances like anabolic steroids, erythropoietin, and human growth hormone, while they can offer short-term benefits, they also raise serious ethical issues by providing unfair advantage and violating fair play. And then they also carry significant health risks. For instance, steroids can cause liver and heart problems. EPO increases the risk of stroke, and HGH may lead to joint pain and abnormal growth. Their unpredictable effects and long-term consequences make them a major concern for both athletes and health professionals. Before we move on to discuss cardiac muscle, please watch this clip that will help summarize the concepts.
Cardiac muscle. The cardiac muscle is located only in the heart. Anatomy. They are made up of striated uninuclear cells joined end-to-end, forming a network. Cell junctions are called intercalated discs. The cardiac muscle has gap junctions. The arrangement of actin and myosin is not as organized as the skeletal muscle. It contains sarcoplasmic reticula, transverse tubules, and numerous mitochondria. The sarcoplasmic reticulum is less developed than the sarcoplasmic reticulum in the skeletal muscle and stores much less calcium. Physiology of the cardiac muscle. It consists of self-exciting tissue. It has rhythmic contractions, 60 to 100 beats per minute. It is involuntary and has all or nothing contractions. It pumps blood to lungs for oxygenation and to the body for distribution of oxygen and nutrients. Excitation-contraction coupling of the cardiac muscle. The action potential spreads out through the cells and across the surface of the sarcolemma via gap junctions. Voltage-gated calcium channels are present in the sarcolemma that get activated. Calcium diffuses into the intracellular fluid from the extracellular fluid. The free calcium present intracellularly activates voltage-gated calcium channels of the sarcoplasmic reticulum. The calcium diffuses from the sarcoplasmic reticulum to the myoplasm. The calcium that diffuses into the cell and the calcium that diffuses through the calcium release channels attach to troponin and initiate contraction.
So cardiac muscle tissue, as we've seen, is found only in the heart, and it's responsible for pumping blood through the circulatory system. Like skeletal muscle, it is striated and organized into sarcomeres, but it differs in structure and function. Cardiac fibers are short, branched, involuntary, and typically contain a single centrally located nucleus. They're interconnected by intercalated discs, which help coordinate and synchronize contractions across the heart. Cardiac muscle tissue has intercalated discs where the plasma membranes of two adjacent cardiac muscle cells are extensively intertwined and bound together by gap junctions and desmosomes. These connections help stabilize the relative positions of the adjacent cells. They also allow a direct electrical, chemical, and mechanical connection between the two muscle cells so that the cardiac muscle cells act as an enormous single cell. And this ability to behave as a single coordinated unit is called functional syncytium. Cardiac muscle cell contractions last longer than skeletal muscle fiber contraction, and this is primarily due to the difference in membrane permeability. Calcium channels remain open in cardiac muscle cells for an extended time, resulting in a prolonged refractory period. Smooth muscle tissue is found in the walls of hollow organs, blood vessels, respiratory and reproductive tracts, in the eyes, and even in the skin. Smooth muscle fibers are short, spindle-shaped, and involuntary, and they each contain a single centrally located nucleus. Unlike skeletal and cardiac muscle, smooth muscle lacks striations, they don't have sarcomeres, or T-tubules, or troponin, or intercalated discs. Here is a short animation of how smooth muscle works.
Smooth muscle tissue is in the walls of hollow organs throughout the body. The tissue contracts and relaxes to contain substances and move substances through the body. Smooth muscle contractions are involuntary actions managed by impulses that travel through the autonomic nervous system to the smooth muscle tissue. The arrangement of cells in smooth muscle tissue provides for contraction and relaxation with great elasticity. The smooth muscle in the digestive tract contracts and relaxes in peristaltic waves that move swallowed food and nutrients from the mouth through the stomach and the intestines. Artery walls include smooth muscle that relaxes and contracts as pressure changes move changing volumes of blood through the vessel. The smooth muscle of the bladder and uterus allow for great elasticity and contraction of those organs.
Smooth muscle contraction is regulated by the calcium-binding protein comodulin, which forms a calcium-comodulin complex to activate cross-bridge formation. Actin and myosin filaments are arranged differently than in striated muscle. The thin filaments attach to dense bodies scattered throughout the sarcoplasm and anchored to the sarcolemma. When contraction occurs, these filaments pull on the dense bodies and intermediate filaments, drawing the ends of the fiber inward and creating a corkscrew-like motion. Smooth muscle fibers communicate via gap junctions in single-unit visceral smooth muscle, allowing electrical signals to spread from cell to cell. Pace setter cells in visceral muscle can spontaneously generate action potentials, coordinating rhythmic contractions like peristalsis. In contrast, multi-unit smooth muscle lacks gap junctions and contracts only in response to individual stimuli. Neurotransmitters are released by varicose rather than traditional neuromuscular junctions. Smooth muscle is highly energy efficient. It's capable of maintaining tone through latch bridges, which allow prolonged contractions with minimal ATP use. It also has the unique ability to grow through hypertrophy and hyperplasia. Hyperplasia is an increase in cell number, and this is seen in the uterus during puberty or pregnancy. Figure 1025 shows the varicosities in smooth muscle tissues. Varicosities are swellings along autonomic nerve fibers that release neurotransmitters into the surrounding tissue. Their vesicles enable widespread diffuse signaling to multiple smooth muscle cells, and this allows for coordinated contraction without direct synaptic contact. All muscle tissue originates from the mesoderm layer during embryonic development. Skeletal muscle cells form when myoblasts, which are embryonic precursor cells, fuse to create large multinucleated fibers, some of which contain hundreds of nuclei. Not all myoblasts fuse. Those that remain become myosatellite cells. Myosatellite cells aid in muscular repair. If damage exceeds the repair capacity of these satellite cells, muscle tissue is replaced by non-contractile scar tissue through fibrosis. Like skeletal muscle, cardiac muscle has limited regenerative ability and is also replaced by scar tissue when injured. In contrast, smooth muscle has a greater capacity for regeneration, thanks to pericytes, are stem-like cells in small blood vessels that facilitate repair. Nerves help define fiber type during development, and physical therapists play an important role in maintaining muscle health, aiding recovery from injury or illness, and designing personalized exercise programs to prevent atrophy and improve mobility.
Skeletal muscles are responsible for the voluntary movement of the body. They are composed of fascicles containing multiple muscle fibers where each represents a single cell. Individual myofibers are covered by a basement membrane, which is continuous with the endomysium connective tissue layer. Peeling away the basement membrane reveals myofibers and the skeletal muscle stem cells, also known as satellite cells, which play a critical role in muscle fiber repair. Peeling away the sarcolemma membrane of the myofiber reveals the intracellular components, including myonuclei, that originate from the fusion of hundreds of myoblasts. The myosin and actin contractile apparatus gives us the ability to move in a controlled manner. Minor trauma events are common in the human body. Following damage to the muscle fiber, nearby quiescent muscle stem cells are prompted to activate and dislodge from their niche. After cell division, the resulting daughter myoblasts differentiate, then fuse with the damaged myofiber and restore its integrity. A major trauma to the muscle can be induced by massive injury, including toxins such as a snake bite. Even after extensive damage, small groups of healthy cells can be found. Following necrosis of the muscle tissue, the remaining viable cells, including muscle stem cells, engage in the regeneration process. The satellite cells activate and generate daughter myoblasts that begin the process of rebuilding the destroyed muscle fibers. During this period, residual debris is removed by the macrophages. The new muscle fibers are generated within the collagen matrix of the pre-existing basement membranes, following the differentiation and fusion of hundreds of myoblasts. Notably, during this regeneration process, satellite cells will self-renew and they are then set aside for future repair events. A range of myopathies can trigger a state of chronic regeneration as myofibers continuously are damaged. As a result, stem cells are frequently mobilized to activate and to produce myoblasts that will repair the ongoing damage to the muscle fibers. Muscle stem cells are quiescent during homeostasis. Following a trauma event, the satellite cells activate, then divide to generate daughter myoblasts that differentiate and repair the damaged muscle fiber. A fraction of satellite cells will self-renew to maintain this stem cell pool. Sarcopenia is the progressive loss of skeletal muscle mass and strength during aging. This process is associated with a decline in the number of muscle stem cells. It is thought that during aging, satellite cells continue to activate, but that the self-renewal process is compromised, resulting in a reduction in the total number of satellite cells over time. This decline in stem cell number, as well as quality, contributes to the loss of muscle mass, quality, and strength during aging.
This concludes the material for Lesson 8. Thank you guys for watching. Don't forget to do the reading, check the description for more videos related to these topics, and leave your questions for me in the comments below.